So let's have a look at the constituents of typical diets and how these things are absorbed. So the majority of our diets contain mostly carbohydrates, depending on if you're on a low-carb diet or not, but most people will eat mostly carbohydrates. So something might be like 300 grams of carbohydrates, 100 grams of fat, 75 grams of ions, and 75 grams of amino acids, which come from proteins in a day, in a typical dietary intake. And you might have about 9 litres of water. Not all of it is drunk, obviously. There's a lot coming from secretions as well, which enters the digestive tract. So before we move on, some features of the small intestine. It's around 3 to 5 meters long, and chyme takes around 2 to 5 hours to pass through it. It has many adaptations which allow it to absorb and pass food through it. So it has mucosal folds of Kirk-ring, which increase the surface area by about 3 times, otherwise known as the valvuli conniventes. It has villi, as we saw in the cross-section, and that increases surface area around 10 times. Microvilli on top of the villi increase surface area by a further 20 times, and these are on the epithelial cells which form a brush border. So the total surface area of the small intestine can be around 250 to 400 meters squared, and this is multiplied by 600 in total. So despite being 3 to 5 meters long, it has a huge total surface area. Now the crypts of Lieberkühn, which we saw were between the villi, the blind ending tubules between the villi, these have several roles. So they secrete fluid, around 2 litres per day, which is matched in volume by pancreatic and biliary secretions, and they contain stem cells, which replace desquamated epithelial cells and replaces the entire epithelium around every 3 to 6 days. The ileocecal reflex is the very last part of the small intestine, where the ileum meets the large intestine, or the cecum. And there's a smooth muscle lining this bit. There's a flat valve from the ileum into the cecum, the appendix here, and this is called the ileocecal valve. So there's 2 reflexes here. When the colon is full, there's increased pressure and decreases the colonal ileal reflex, so food slows down here. But when the stomach is full, there's a gastroileal reflex, which increases the rate of movement through here by opening this valve flap. The motility in the small intestine is controlled by slow waves, and this is how they vary. So this is the membrane potential against time. In the proximal part of the gut, you can see there's many slow waves, and in the distal part of the gut, there's not that many slow waves. So about 12 per minute in the duodenum, decreasing to around 8 per minute in the terminal ileum. Contraction requires large enough slow waves to generate action potentials, so they are important in the motility of the small intestine. There's 2 types of motility in small intestine, mainly, which is similar to other parts. So there's segmentation and peristalsis, and segmentation is the main form of motility in the small intestine. It's weakly propulsive and involves churning and mixing. Peristalsis involves moving a short segment at a time, modulated by gut wall stretch and hormones. The contents move into the large intestine when the terminal ileum is distended, and the enteric nervous system causes a peristaltic reflex, which causes this to happen, along with the gastroileal reflex, of course. So protein digestion, we said that 75 grams of amino acid, around that amount, is consumed per day. How is this all digested? So protein enters the digestive tract, and firstly, it's acted on by pepsins. So around 15% of the digestion occurs by pepsins, and pepsins are especially important for breaking down collagen because they can't be broken down by these other enzymes. And hydrochloric acid also helps break down and denature the proteins. When they're polypeptides, they move into the small intestine, and on the brush border of the small intestine, there's a lot of enzymes which break down amino acids. There's trypsin, chymotrypsin, elastase, and carboxypeptidases, and many more. Trypsin is activated by an enteropeptidase from the inactive form trypsinogen, and the presence of trypsin itself increases the activity of enteropeptidase, so it self-activates. It's a positive feedback loop on the brush border. And these other enzymes are also activated by trypsin, so these are pancreatic proteases. These are chymotrypsin, elastase, and carboxypeptidases, which break down polypeptides into peptides. And these peptides then are processed by brush border peptidases, which catalyze the breaking down of the peptides, the chains, into ditripeptides and amino acids. So these brush border peptidases are in the upper small intestine on the epithelial cell apical membranes. Now these different types of products, the ditripeptides and amino acids, they all enter a small intestinal cell via their respective channels, and they can be interconverted into amino acids and glutamine, which can be stored inside, or amino acids which are released. And this all happens via facilitated diffusion and secondary active transport, so transporting them against their gradient with an ion. And then the amino acids are transported out into the blood. Some amino acids like glutamine are oxidized for energy in the rapidly dividing intestinal cells, so they directly use these instead of transporting them. Starch, carbohydrate digestion, so we said around 300 grams, so most of the diet was starch or carbohydrates, and 50% around is starch. So starch enters our digestive tracts, and then salivary and pancreatic amylases begin to act on it. It cleaves internal alpha-1,4 bonds, so the typical bonds between your monosaccharides, but it can't cleave any branching bonds, so branches from 1,6, it can't cleave those. And starch is then processed by these amylases into oligosaccharides and alpha-dextrins. Alpha-dextrins contain 1,6 bonds which can't be broken down. So then these oligosaccharides and alpha-dextrins reach the duodenal or jejunal brush border, and this is a gut epithelial cell which contains many enzymes in its membrane, so it has a glucoamylase, which, similarly to the salivary and pancreatic amylases, cleaves 1,4 alpha bonds. It has alpha-dextrinase, which can cleave the branching bonds, so it can break down the branched ones which have alpha-1,6 bonds, and sucrase and lactase, which break down their respective disaccharides. So sucrase breaks down sucrose into fructose and glucose, and lactase breaks down lactose into galactose and glucose. So then these are processed and broken down into their monosaccharides. Now, the small intestinal epithelial cell absorbs these glucose and monosaccharides. So in the lumen, we have the broken down products, and then the glucose and galactose enters through SGLT1 transporter in combination with sodium, which uses the ion gradient to transport it, and fructose enters through a GLUT5. And these can be interconverted all into glucose and exit through GLUT2. And this whole thing is powered by a potassium-sodium transporter, ATPase, unsurprisingly on the basolateral membrane, which causes an ion gradient for the sodium to move in. Ion absorption also happens in the small intestine. So iron absorption, Fe, that is, it happens in the duodenal cells, and calcium absorption happens in the small intestine. So calcium and iron availability is decreased by binding other dietary constituents. For example, gastric acidity has to increase the solubility because of this, and phthalate ions can produce insoluble salts which can't be absorbed, so that's not very good. So it's important that these things are absorbed because they're important for function in our body. In the duodenal cells, this is the lumen, this is the extracellular fluid, we have iron in the form of Fe3+, which can't be absorbed. It has to be reduced by either vitamin C or iron reductase enzymes on the duodenal cell membranes. Once it's reduced, it can enter the duodenal cell via a DMT1 transporter, which transports it along with a hydrogen ion, or it can enter as heme, so bound to protein. Then the iron is all isolated, and it can either bind a protein and become ferritin, or bind other proteins and be transported out through this ferroportin, and then once it's transported into the extracellular fluid, it's oxidized again to Fe3+, and is bound to transferrin protein, and then is transported into the blood. And the thing about the ferroportin protein is that its activity can be modulated by a hormone called hepcidin from the liver. So this is a peptide hormone which increases the response to inflammatory mediators and decreases the blood iron levels. So what it does is it decreases the activity of ferroportin, so less iron is transported out, more iron is converted into ferritin and stored inside this cell, and then these cells are often replaced, so the iron is lost that way. Now, moving on to calcium absorption in the small intestine. So calcium absorption is dependent on vitamin D, which is why it's important to get out into the sun. So this active form of vitamin D, 1,2,5,2-OH-D, acts on the nucleus, which then transcripts and translates, and the RNA produces proteins which increase the activity of these transporters and their numbers. So what happens is calcium in the lumen is paracellularly absorbed along its gradient and also through a transporter entering the cell, where it binds to calbindin, and then the calbindin calcium complex then allows the calcium to be transported out through ATPases or through a calcium sodium pump. And this is all powered by a ATPase NAK pump as well. And the vitamin D increases the rate and the number of transporters here. So it's more reliant on paracellular calcium uptake if the uptake is very high, but if the uptake is low, then active vitamin D acts on the receptor in the nucleus and increases the regulation and transcription of transcellular uptake proteins. Other ions that need to be absorbed include sodium, potassium, chloride in the small intestine and large intestine, and they are absorbed slightly differently. So in the small intestine, which is mainly the jejunum here, we've got the lumen, extracellular fluid, so we've got our basolateral pump, which is providing a gradient, and the activity of this is increased by aldosterone. And in the small intestine, we have a KCl pump on the basolateral membrane as well. And chloride is being absorbed paracellularly as well as through the cell. So here we have a bicarbonate chloride transporter, which switches a bicarbonate for chloride, and then a sodium hydrogen transporter, which absorbs the sodium as well. Sodium is mostly absorbed in the small intestine. It's coupled to monosaccharides and amino acids, like glucose and amino acids, through SGLT, and also sodium hydrogen antiporters and the ENaC in the large intestine. And in the large intestine, things are slightly different. So we still have this chloride bicarbonate transporter and the sodium hydrogen transporter, and then we also have this pump here, but then we have an ENaC which exchanges potassium for sodium. And chloride is absorbed as well paracellularly, and also by exchange with the bicarbonate. There's not much K+ absorbed actually, there's net K+ secretion, but most of the secretion and absorption is paracellular. And aldosterone increases the activity of the pump in the large intestine as well, and this arrow is supposed to be pointing to the ENaC, so it increases the ENaC activity. Those are how those three important ions are absorbed. Water is important to be absorbed as well, and the way that it happens from our food, so we can absorb water by drinking it in the kidneys, or we can absorb it from food. This happens mostly in the ileum, but some of it happens in the colon depending on how much water in the food there is. These are ileum epithelial cells, and they are pumping out sodium into this tight junction here between them and pumping in the potassium through ATPase transporters. And that's increasing the ion concentration as well as increasing the concentration of chloride. So the water will move down its concentration gradient via osmosis through this partially permeable membrane as well, and that increases the water pressure here where there's a lot of ions. And the pressure pushes the water into the capillary through the basolateral membrane. So the increase in concentration of ions drags water in, and the increased water pressure pushes the water into the capillary and absorbs it into the blood. This is isoosmotic, even though you would think that there would be a higher concentration of water out here because the small intestine epithelium is very leaky, so there's a pretty much equal concentration of water and ions on both sides. There's decreased water absorption in the colon, but increased osmotic gradient because tight junctions between cells are tighter and limit the back diffusion of ions. So this bit is tighter in the colon and there's less back diffusion, so that the gradient is even higher. So just some figures here. Water enters the digestive tract around 2 litres per day from ingestion, so food, and around 7 litres per day through gastrointestinal secretions. So that makes up the majority of the water in the digestive tract actually. The small intestine, especially the ileum, absorbs around 7.5 litres, and the colon absorbs around 1.4 litres, but it can take 2 to 3 times more if necessary. And around 100 millilitres of water is lost in the feces. Now the liver. So the liver is an important organ with many functions in the digestive tract. It's made of lobules, so if we zoom into one of the livers, it's not quite cells, this is a liver cell, but this is a functional unit of the liver, so it's made of lobules, and this is a portal triad. So there are many hepatic vein branches going through the liver, and these all connect to hepatic sinusoids. So this portal triad would be here at every corner of this hexagon, but I've only drawn one. So these lobules are around 2 millimeters diameter, and so this distance would be around 2 millimeters from one portal triad to the next. And the portal triad contains a hepatic artery, portal vein, and a bile duct. So the blood is going from the artery and the vein to the hepatic vein, and the bile is going in the opposite direction, so it's a countercurrent flow into the bile duct through the bile canalicculus, and this is mixed blood. And what happens here is that there are many liver cells surrounding the blood, detoxify the blood, they help metabolize substances and other functions that we'll look at later. So a liver cell here, we're looking at metabolism primarily. So as the blood moves through, the glucose, fructose and galactose and all the other sugars will be absorbed through the GLUT2 and converted into glucose, or used up by the liver cell. But most of it is converted. And also lactate, amino acids and glycerol will be converted into glucose via gluconeogenesis. Glucose can be stored as glycogen in the liver cells, or converted into triglycerides and then exported as VLDL, very low density lipoproteins. If they're all converted to glucose, then the glucose is transported out through a GLUT2 protein as well, on the other side into the blood as glucose when we're fasting. So functions of the liver now, there's 6 main functions. So one function is, obviously as we saw, carbohydrate, lipid and protein metabolism. So the liver takes up monosaccharides from the portal vein via GLUT2 facilitated diffusion. Glucose is converted to glycogen, around 100 grams in liver, 400 grams in muscle cells with a 24-hour supply. So most of the glycogen is stored in muscle cells, but some is also stored in the liver. Lactate and amino acids like alanine and glycerol are converted into glucose, and gluconeogenesis occurs when carbohydrate stores are low. Intercellular proteins are decomposed via lysosomal enzymes, amino acids enter the blood. Extracellular proteins are digested by macrophages. Most amino acids are recycled, around 30 to 50 grams are broken down per day for energy, and excess amino acids are oxidized for energy or converted to glucose and ketone bodies. The liver interconverts amino acids, pyruvate, TCA cycle, same thing as Krebs cycle, enzymes by intermediates by transamination. And excess glucose is converted into triglycerides, exported as lipoproteins and stored in adipocytes as fat. Another metabolic function of the liver is to secrete VLDLs when fasting, so this is the lipid side of things. It exports triglycerides and hepatic cholesterol. VLDLs contain phospholipids and apoproteins like chylomicrons, and lipids are oxidized into ketoacids, so things like acetoacetate, beta-hydroxybutyrate and acetone, which are ketoacids. And the brain uses around 75% of energy from ketone bodies, but it always has to use glucose. Second function of the liver is bile formation. The bile is the excretory product of the liver. It's composed mostly of liquid, contributed by the bile duct epithelium. The 65% bile acids, so these are containing salts of cations, for example sodium salts, and conjugated with glycine or taurine, so this increases their solubility. Primary bile acids are made of cholesterol in the liver, and gut bacteria also help make secondary bile acids. It's 20% phospholipids, 4% cholesterol, and a little bit of bile pigments as well, which make it green. The roles of bile are, number 1, to promote fat absorption, because it contains surfactant working with phospholipids, so these have a hydrophobic and hydrophilic side to them, so they act a bit like an emulsifier and help absorb the fat, because fat is not soluble in water. Also, it helps with waste excretion, so cholesterol and heavy metals such as copper and cadmium, and it's important for protection and defense. So, for example, immunoglobulin A, mucus, and tocopherol antioxidants are contained within bile and help to initiate an immune response or stop bacteria, germs, pathogens getting further down into the digestive tract. It also stores vitamins, destruction and detoxification of hormones, drugs, and toxins, so metabolism of things that we don't need and toxins, and blood filtration, so removing effete erythrocytes, that's dead red blood cells, and shoving bacteria into the hepatic portal vein, so getting it out of the blood. And it acts as a blood reservoir as well, because the splanchnic circulation, the hepatic circulation, is very compliant, and the hepatic vasoconstriction empties more blood into the circulation.